mapping, right? We are going to understand about three types of mapping. One is called as conjugation mapping, other is called as transformation mapping, and the third one is known as the transduction mapping, right? Now, before moving ahead, we should be familiar with these basic terms as well. I mean, what is the concept that you need to understand? You need to know about conjugation, you need to know about transformation, you need to know about transduction as well. So I'm going to begin with brief definition of these terms. So Vignesh, can you try to define conjugation now? How to define the word conjugation?
Genetic exchange between two bacteria with the physical contact.
Okay, so maybe we have to define the direction also. Can I say it's a unidirectional
Yes.
transfer of genetic information? So quickly write everyone, unidirectional transfer of, so unidirectional transfer of genetic material through direct cellular contact, through direct cellular contact between a donor and a recipient bacterial cell, between a donor and a recipient bacterial cell. So unidirectional transfer of genetic material through direct cellular contact between a donor and a recipient bacterial cell, right? I hope you understand. So there's one donor, there's one recipient bacteria, and transfer is always going to happen from the donor to the recipient bacteria. That's why we are using the word unidirectional transfer, right? Hope you understand about that.
Yes, sir.
Now, how do we decide on what presence or absence do we decide that whether one bacteria would be called as a donor bacteria or not? So the donor state, I hope you agree with this point. The donor state is conferred to a cell. The donor state is conferred to a cell based on the presence of based on the presence of a special plasmid. So based on the presence of a special plasmid, which is called as an F factor, right? Based on the presence of a special plasmid, which is called as an F factor. I hope you have heard about it. So based on the presence of a particular thing called as F factor. I mean, there is a particular bacteria in which you can see that there's the main chromosome only, right? You have the main chromosome only. This can be considered, I mean, to be your F minus factor, right? F minus consider kar sakte hain. So if there's a main chromosome only, you will call it as F negative, right? And if the bacterial cell, right? If you look at the bacterial cell and that bacterial cell is having the main chromosome, it is having the main chromosome as well as a special plasmid, which is called as the F factor, right? So F factor is also present based on that fertility factor. This is called as F plus. So I hope you understand. This is called as F plus would be considered as donor in our upcoming numericals today. F minus are called the recipient, right? I hope that makes sense to everyone. So on what basis donor and recipient is decided? If F factor plasmid present, right? So F factor is nothing but a special kind of plasmid. If that is present, then it is called as your what? If donor bacterium or the F positive, and if the only main chromosome is there and F factor is absent, then it is called the recipient bacterial cell, and that is going to be the forming the basis of our conjugation mapping. Is it clear or not?
Yes, sir.
Is it written till here? Okay. So I would like to add few more things over here. यहाँ पर थोड़ा सा add करेंगे जरा एक important information. So what I'm trying to say is that the conjugation always results. So conjugation always results in unidirectional transfer. Conjugation always results in the unidirectional transfer of a copy of the F factor, from the F plus to the F minus cell. That is from the donor to the recipient cell, right? From the donor to the recipient cell. So conjugation always results in a unidirectional transfer of a copy of F factor from the F plus, that is donor, to F negative, that is the recipient cell, right? So is that clear to all of you?
Yes, sir.
Okay. Shall we move ahead after that? I hope it is written by everyone. Please थोड़ा सा quickly लिखेंगे हम.
सर ये प्लाज्मिड सभी बैक्टीरिया में नहीं होता क्या? मतलब ऐसे
प्लाज्मिड
कुछ
सभी
उसमें
बैक्टीरिया में
भी
नहीं होता
मतलब जैसे यहाँ पे तो अपन ने मतलब F1 सॉरी F फैक्टर है
हाँ
या
जी.
प्लाज्मिड है, इसके बेसिस पे अपन ने डोनर को डिफाइन किया है.
हाँ हाँ, सबके
बट
पास
सर
नहीं
ऐसे
होता.
भी तो
सबके
होता है.
पास होता तो सब डोनर नहीं हो जाते कुलदीप.
नहीं सर, कुछ बुक्स में ऐसे लिखा होता है ना कि मतलब बैक्टीरिया में मेल क्रोमोसोम के अलावा प्लाज्मिड भी प्रेजेंट होते हैं.
तो भाई प्लाज्मिड प्रेजेंट भी हो सकता है, एब्सेंट भी हो सकता है. प्लाज्मिड जिंदा रहने के लिए जरूरी नहीं है. बैक्टीरियल सेल में से प्लाज्मिड हटा दोगे तो बैक्टीरिया मर नहीं जाएगा. आदित्य सर ने भी पढ़ाई है ना ये बात. बैक्टीरिया इज नॉट गोइंग टू
नहीं
डाई.
सर, बस मैं ये
हाँ
इतना ही पूछ रहा था कि जरूरी थोड़ी है कि सब में हो, कुछ में एब्सेंट भी तो हो सकता है ना
हाँ, तो
प्लाज्मिड.
इसीलिए तो ये लिखा ना. एफ नेगेटिव में कहाँ है प्लाज्मिड, बताओ कहाँ है प्लाज्मिड. इसमें
नहीं
मेन
सर.
क्रोमोसोम ही तो है बस, है ना.
यस सर.
राइट,
यस सर.
समझ गए. तो आप सबने लिख लिया है तो थोड़ा सा स्पीड बढ़ाएंगे लिखने का भी. देन ओनली विल बी एबल टू मूव अहेड विद दीज टॉपिक्स, राइट. कैन आई मूव अहेड आफ्टर दैट?
Yes, sir.
कुलदीप लिख लिया ना? कुलदीप, इज इट डन?
यस सर.
नाउ देखिए, there is one more situation which you should be familiar about, called as HFR strains, right? There are certain strains in bacteria called as the HFR strain. So one is F negative, one is F positive. What happens in HFR strain? So in HFR strain, you will see a situation like this. So HFR में क्या होता है आपका, that consider that this is your main chromosome. In your main chromosome, portion of the F factor gets integrated, right? So then such strain would be called as what? So they would be called as HFR strain. So this is your main chromosome part, main chromosome. But in main chromosome, a part of F factor get integrated. So F factor is not a separate plasmid now. So in HFR strain, what has happened? So if the F factor gets integrated, if the F factor gets integrated into the bacterial chromosome, so if the F factor gets integrated into the bacterial chromosome, then such strains are called as, so जो strain ये possess कर रहे हैं, right? So such strains are known as, HFR strains. तो ऐसे strains को क्या बोला जाता है? HFR strains कहा जाता है. I hope that makes sense to everyone. So if the F factor gets integrated into the bacterial chromosome, then such strains are known as what? HFR strains, right? I hope that makes sense. Is that clear or not?
Yes, sir.
यस सर.
अच्छा, अब दूसरी बात यहाँ पर है कि वेरी इंटरेस्टिंग थिंग दैट यू आर गोइंग टू अंडरस्टैंड ऑल्सो. आई मीन, what happens is that if an HFR strain, right, if an HFR strain forms a conjugation tube, right, this is an HFR strain, I mean something of this sort. When we are representing, let's say I'm instead of double stranded, I'm simply making single stranded for just saving time, right? Do not understand that it is like this. So what happens is this thing during conjugation, what we actually do in conjugation mapping, what we try to decode over here. So if this is an HFR and it is conjugated with F negative, so let's say the genes over here are A plus gene, some genes are like B plus, C plus. I mean, if you talk about the F negative, its main chromosome should not be having these genes A minus, B minus, C minus, like situation like that. So when the transfer begins, right? So what we are actually interested in conjugation mapping, right? Just be very careful about this thing. All conjugation mapping numerical will start with a statement that there is a conjugation between HFR and F negative, right? And I mean, whatever genes are getting transferred, right? So what we can do is, with the help of conjugation mapping, you can do one thing. You can easily determine, so you can easily determine, two aims are there. You can easily determine correct sequence of genes, correct sequence of genes, correct sequence of genes on HFR chromosome, right? Correct sequence of gene you can easily determine, just like in linkage you used to determine A plus. There's a point over here from where transfer happens, right? There's a point like this which is called as origin of transfer, right? Origin of transfer, and with that respect all distance are measured, just like centromere or used to be there in eukaryotic system. So let's say this is the origin of transfer, right? This point is the origin of transfer because all distance are measured with respect to this thing. I'm just giving brief hint what we will be doing in conjugation mapping. So whether the correct order is A plus, B plus, C plus, right? I mean the nearest gene, right, to this point, or it can either be B plus, A plus, uh, so we can have B plus, C plus, or A plus, right? So there are multiple possibility. You have to find out the correct sequence, and one correct gene order is there, right? Once you have established the correct gene order, what are you going to do, Vignesh? We can also find out distance between these genes, right?
Yes.
There are two main aims. Acha, what was the unit of distance measurement in eukaryotic mapping? What was the unit in eukaryotic genetic mapping? What was the unit of eukaryotic genetic mapping?
Centimorgan.
Centimorgan or map unit. But in bacterial mapping, all distance are measured in minutes. All distance are measured in minutes. The unit is minutes, right? This 10 minutes. I mean, how we are measuring in minutes? Let's say after 10 minutes, the F negative started producing A plus protein. Then what would have been the distance of A gene from origin? It would have been 10 minutes. That's how we measure the distance, right? I will tell you everything. Distances are measured in minutes in bacterial mapping. Is that clear or not? That is going to be the main aim of conjugation mapping. अभी सिर्फ नोट करना है, ये बहुत डिटेल में आने वाला है. Do not get confused. Is the main aim clear to you? So determining correct sequence of gene on HFR chromosome with respect to origin of transfer, and then finding the distance between the genes in minutes. Does that make sense to everyone? This is a brief thing what includes in conjugation mapping. I'm moving ahead towards the definition of transformation. Bollier ji, is it clear or not? Done. I'm removing the information now.
Yes, sir.
So next that we have is, the next thing is second one, which is your transformation mapping, right? I mean transformation mapping के लिए हमें transformation term we have to understand. So transformation is nothing, I hope you understand. I mean there's a particular cell, right? And in its, I mean in solution form surrounding that cell, or in aqueous solution, right, you have some pieces or fragments of genetic material which are lying in the surrounding, right? So the natural uptake, right, of those fragments of genetic material, or let's say uh, extracellular pieces, right? We can say extracellular pieces of DNA, right? extracellular pieces of DNA, right? Extracellular pieces of DNA इसके आसपास present है, right? So how we define transformation? So it is the transfer of genetic material, transfer of genetic material, between organisms, right? Between organisms by small extracellular pieces of DNA, small extracellular pieces of DNA, right? Small extracellular pieces of DNA. So let's say extracellular pieces of DNA belong to bacterium 1, right? Let's say donor bacterium ka, these are the things. Let's say they belong to donor bacterium. Donor bacterium has been fragmentized, right? It has been completely destroyed, and let's say this particular bacteria which is going to receive it is your recipient bacteria. Here also we have terms: recipient bacteria, donor bacteria. Let's say it got completely fragmented, right? Bacterial cell completely damaged. Then all the genetic material would be present in the solution form that has to be uptaken by the recipient bacteria. Don't worry, this is just basic definition. I'll show you what thing is being done on the mapping. Is that clear or not? Now one more thing I would like to draw for you. Suppose there's a recipient cell, right? I mean, what happens in the transformation mapping, there's a recipient cell. Let's say it is A negative, B negative, C negative, just like this, just giving one example, right? And there's a fragment, right, which has just been received. This is a fragment coming from donor bacteria which is having A plus, B plus and C plus, right? What happens in transformation thing? Because of the recombination event, right? So यहाँ पर what happens is crossover event, right? Crossing over event. What will ultimately happen? These particular fragments actually get embedded. So this particular red fragment is your fragment from donor bacterium, right? Your donor fragment, I might write it like this. So this is our donor fragment like this, and what ultimately happens over here is something of this sort, right? So what happens over here is this particular thing. I mean, this much section which is present over here, this will become A plus, B plus and A plus, B plus and C plus हो जाएगा यहाँ पर, and this is going to become your A negative, B negative and C negative, right? So once again, what is the purpose? What do you think can be the purpose of the transformation mapping? It can actually help us to define. So if we talk about transformation mapping, what kind of numericals you'll be solving in the lectures? So in transformation mapping, our main aim, if this is a donor fragment, right? If this is a donor fragment, we would be possibly able to determine that what is the distance between these genes, right? So what is going to be the distance, right? We cannot say the order because we cannot define where the origin of transfer is, but since it is a random fragment, the main purpose over here will be to determine the distance between the genes, determine distance between these linked genes. Since they are linked together, that's why they are always having a chance to be transferred together. So the distance between the linked genes, once again, these distance also going to come in minutes, because in bacteria, never use the word map unit or centimorgan. Always distance should be coming in the minutes. Is the brief aim of transformation mapping now clear to you? But we can define one thing. If we cannot determine where the origin of transfer is, we can still decide which is the central gene. That makes sense to you or not? अरे बोलो यार, please be responsive.
Yes, sir.
ठीक है.
Sir, can you repeat what you just said?
In last transformation mapping, right?
Yes, sir. No,
ठीक
central
नहीं,
gene use.
हाँ, transformation mapping में will have to this particular thing could also have something like this: C plus over here, A plus over here, and B plus. I mean, let's say B plus over here, and A plus in center. I mean, still we can decide. Although we cannot, there's no need to determine the correct order because there are no ends which are over here, right? There is no centromere over here. This is just a random fragment, right? Random fragment containing but 3 linked genes, right? Among these 3 linked genes, can you still possibly try to determine the central gene, right? We will learn method for that, right, today.
Okay.
And then once we determine central gene, we can find out the distance between this, we can find out distance between this, and therefore the total distance between these. All distance will be coming in minutes, right?
Yes, sir.
Right, Vignesh? This is just a brief intro. You will now be landing up into the world of individual mapping after that, right? And where numericals will be coming.
Okay, sir.
Does that make sense to everyone?
Yes.
ठीक है. So यहाँ तक आपने अगर लिखा हो तो बताइए जरा. Is that done? फिर ट्रांसडक्शन के बारे में जरा एक बेसिक डेफिनेशन. Then we'll be moving ahead with the other basics after that.
Yes, sir.
Okay, ठीक है जी. Moving ahead after that. So transduction, right? Talking about transduction. One word definition. So let's say, so bacteriophage mediated gene transfer, bacteriophage mediated gene transfer from donor bacteria to recipient bacteria. Okay, so bacteriophage mediated gene transfer from the donor bacterium to the recipient bacterium, right? So from donor bacterium to the recipient bacterium. So bacteriophage mediated gene transfer from donor bacterium to the recipient bacterium, right? So once again, what we have over here is, let us suppose, just giving you a brief overview, quickly make these figures. These figures will come once again at a later stage when I will start transduction mapping. So there's a donor bacterium, right? Let's say this donor bacterium is having three genes like this, A plus, B plus, C plus, right? Let's say it is now attacked by a phage, right? It is being attacked by a bacteriophage, right? So attacked by a particular bacteriophage. I hope all of you understand that when this particular thing is being done, I mean attack is being done within this donor bacterium also during the infection cycle. Do you know that there will be fragmentization of the genetic material? Right? Fragmentation of host genetic material also happen because this is a part of the damaged virus life cycle, right? Fragmentation of donor chromosomes or donor material, right? So, and if these three genes are tightly linked, there is highly chance that these three genes might also come on one fragment. Does that make sense? अगर ये donor chromosome पर काफी पास रहे होंगे or they would have been strongly linked, let's say we write that they are linked together, then it is quite possible then when fragmentation would be happening randomly, these three fragments may land up on same fragment also, right? Or maybe two can be on one, other one can be on other fragment, but I'm just giving you examples. Is it clear or not? Then what happens the next thing after that? Lysis of the host bacterium. Bolo bhaiya. Lysis of the host bacterium is there. Everyone agrees to that. So lysis of the host bacterium is there. And do you understand that? Let's say this show this thing as viral RNA. Viral RNA, right? So what is going to happen? That I hope everybody is clear with this fact that progeny viruses will be produced once lysis is done.
Yes, sir.
Progeny viruses are this thing, right? Now there are two types of things. One is called as the normal phage progeny, a normal phage. And how do you decide if it is a normal phage? It is a normal phage if it is containing viral RNA inside it. If it is containing viral RNA, right? But don't you think that by chance it is also a possibility that it might pack one of the fragments, any random fragment of the donor bacterium also, right? Because that is also fragmentized. Let's say by chance, by mistake, this donor fragment is there, then it is called as defective phage, right? What it is called as?
Defective phage.
Why it is called as defective phage? Because it is containing a part of the donor chromosome. यहाँ तक समझ आती है बात या नहीं, बताइए पहले. Normal phage means if during packaging inside its viral head it receives its own RNA, right? Because when the virus enters and attacks, there are also RNA copies which are present inside it. Its own genetic material is also there. And once packaging starts, viral RNA will be packed inside viral head to form new progeny phages. But it is quite possible that a defective phage may also be generated, right? Do you digest this fact or not? And now let us see what will happen if this defective phage is going to attack. If this defective phage is going to attack a particular recipient bacteria, this is going to attack a particular recipient bacteria, which is A minus, B minus, and C minus. And when it is going to attack, let's say, so this defective phage has attacked at this particular point. So once it is attacked, it will push this RNA in front of this, which is once again going to be A plus, B plus, C plus, right? And once again, there will be a crossover event. And finally, what you'll see over here is that in the recipient bacterium, this particular thing has become a part of this thing. So you can write like this thing over here, which is A plus, B plus and C plus like this. ये यहाँ पर आ जाता है आपका. And you have this particular segment which is A negative, B negative and C negative. Once again, based on this particular mapping, this type of mapping, you can determine or try to know about central gene information. Central gene information can be easily determined, and we can also try to deduce about the gene distances, right? Gene distance in minutes. Gene distance in minutes, right? Bolije, is that clear or not?
Yes, sir.
Yes, sir.
Okay, let me tell you one thing. If you're talking about transformation, right? I mean, just giving you one concept. If you're talking about transformation, I mean, you'll hear terms like that. And let's say there are three genes like this, A plus, B plus, C plus, and they are transferred to recipient, right? They are transferred together to recipient, transferred together to recipient, right? So there's one term that you'll hear in the numericals. It is called as co-transformation. What it is called as? Co-transformation. What is the meaning of co-transformation? Together ke liye we are using the word co-transformation. So the A, B and C are getting co-transformed, meaning they are together transferred. Therefore, that means they can be present linked, right? So when together something gets transferred, we are using the word co-transformation. And if same term you apply for transduction, this is called as co-transduction. Co-transduction, right? Since they have been transferred together, this event is called as co-transduction. Such terms would be coming in the numericals, right? Are you clear with this term? You cannot use such words for conjugation, right? Conjugation में तो transfer is happening step by step, right? Exactly, the genes are getting transferred from F plus to F negative. But for transformation, whatever genes are landing on one fragment and getting transferred from donor to recipient, and same goes for transduction also. Co means together, right? So we use the word in numericals co-transformation and co-transduction. Is that clear or not? That's the basis of our conjugation mapping. Does that make sense to everyone?
Yes, sir.
Sandhya, is that clear? Are you there?
Yes, sir.
चलिए, okay. So can I move ahead after that?
No, sir.
Yes.
अच्छा, लिखो एक बारी.
Done, sir.
Is it done? Okay.
Yes, sir.
Yes, sir.
Moving ahead after that, there will be some basic terms that will be utilized. Some more basic terms would be there. Some basic terms required for mapping studies, and we'll quickly be understanding about those. And then we will start with first topic for today, which is conjugation mapping. So the basic term that we need to understand, one of these terms, which is called as what? Minimal media, right? So we have one of these terms called as what? Minimal media. So we call it as minimal media. So how to define minimal media? So media which contains the nutrients which contain nutrients required for growth of wild type cells. Required for, so which contain nutrients required for the growth of wild type cells. So media which contain the nutrient for the growth of wild type cells is called as what? Minimal media. इसे क्या बोलते हैं आपका? Minimal media कहा जाता है. For example, I hope you understand. Let's say we talk about minimal media. So let's say minimal media for wild type E. coli. This is quite important. Wild type meaning which can synthesize each and everything by itself also. So minimal media of wild type E. coli consist of few basic things, right? Consist of a sugar. So consist of a sugar as a carbon source. Consist of a sugar as a carbon source and some salts and trace elements, right? And some salts and some trace elements. So from minimal media, the organism can synthesize. So from the minimal media, the organism can synthesize all other components. It needs for growth. All other components it needs for growth and reproduction. Right? Including amino acids, vitamins, etc. Right? So the main thing is what I'm trying to say. If a bacteria is of wild type का bacteria है तो minimal media में बस sugar, some salts and trace element are there. By rest everything bacteria can synthesize since it is a wild type bacteria. So wild type bacteria you can easily grow on minimal media, which contain basic things only: sugar, some salts and some trace element. Rest bacteria can synthesize everything because it is not a mutant, because it is a wild type. I hope you must have heard some terms also. So wild type strains are also known by this word. So prototropes or wild type, right? Such type of terms will appear in the questions today, right? So prototroph or wild type. I hope that makes sense to everyone.
Yes, sir.
So prototroph और wild type की यहाँ पर बात की जाती है, right? Prototroph और wild type. So does that make sense to everyone?
Yes, sir.
मिनिमल मीडिया में कोई दिक्कत नहीं है. फिर सेकंड टर्म जो क्वेश्चंस में अपीयर होगा, इज इट रिटन एवरीवन? सेकंड टर्म है हमारा कंप्लीट मीडिया. तो सेकंड टर्म दैट वी आर गोइंग टू हैव, प्लीज थोड़ा सा तेज लिखिए, है ना, अभी बहुत सारी चीजें देखनी है. फिर कंप्लीट मीडिया के बारे में बात करेंगे. व्हाट इज कंप्लीट मीडिया? which supplies vitamins, amino acids and all other essential metabolites apart from the basic requirements like sugars, etc. Right? So complete media, it is necessary for the growth of mutant strains, mutant/auxotrophs. So prototrophs or wild type and mutant/auxotrophs, one and the another word that you have commonly heard about, right?